The snow leopard dictates the population dynamics of high-altitude ecosystems across the Himalayas. As an apex predator, its hunting patterns shape the survival of both wild and domestic species. To understand these patterns, researchers focused on 3 rugged regions in Nepal: Lower Mustang, Upper Manong, and Sagarmatha National Park. Historically, biologists tracked this predator's diet by walking linear transects along cliff edges and ridgelines to collect scat samples. Relying solely on these field samples creates a critical vulnerability. Most historical data detailing snow leopard trophic ecology is built on tracking methods that are highly prone to misidentification. Correcting this ecological model requires a rigorous 3-step methodological overhaul. We have to apply genetic verification to the samples, convert the findings into relative biomass, and cross-reference that data against an environmental availability index. Failing to apply these filters leads to inaccurate dietary profiles, which misdirects conservation policies and wastes resources protecting a predator based on faulty assumptions. The initial data collection requires immense physical effort. Field teams surveyed over 139 kilometers of high-altitude trails, gathering hundreds of putative scat samples. Here, researchers encountered the first major point of failure. To the human eye, scats left by sympatric mountain predators like the Tibetan wolf, red fox, or common leopard look identical to those of the snow leopard. To bypass this visual illusion, researchers extract DNA from the samples and amplify a segment of mitochondrial cytochrome B using PCR genotyping. This acts as an absolute biological filter. The DNA tests showed that exactly half of the collected samples did not belong to snow leopards. 50% were left by other carnivores. Because early studies rarely use genetic markers, this 50% error rate silently contaminated decades of literature on snow leopard diets. Without establishing a genetic baseline first, any resulting ecological model is heavily distorted by sympatric carnivore noise. Once the snow leopard samples are verified, researchers wash them to extract undigested bones, teeth, and hair to determine what the animal actually ate. This image shows the microscopic morphology of 2 different hair samples. Researchers use these unique cellular structures to identify the exact prey species. Notice the right side of the image. The specific overlapping cuticle scale patterns allow scientists to differentiate between closely related prey types. Now look at the left side. Analyzing the dark, thick medulla structure running through the center of the hair provides the definitive metric needed to separate a domestic yak from a wild blue sheep. Applying this dual-layered rigor, first verifying the genetic origin of the scat, then identifying the microscopic prey structures, turns an ambiguous sample into an undeniable ledger of predatory behavior. Historically, researchers quantified this diet using a metric called relative frequency. It simply counts how often a specific prey species appears across all the scat samples. This creates a statistical trap. Relative frequency heavily overestimates the dietary importance of animals like pikas and voles. A snow leopard might eat many of them, but they yield very few calories. To map true caloric intake, biologists must use a corrective metric known as Relative Biomass Consumed, or RBC. This calculation converts raw frequency into an accurate measurement of energetic reliance by factoring in the average body weight of the consumed prey. This chart compares relative frequency against biomass consumed. When we apply the RBC equation, the reliance on small mammals drops to near 0, while the contribution of large prey spikes. Specifically, the data reveals that domestic yaks provide 44% of the overall consumed biomass, emerging as the dominant caloric source. Frequency metrics only map how often a predator hunts. Biomass metrics reveal the true energetic foundation keeping them alive. This raises a central analytical paradox. If yaks supply nearly half of their caloric intake, are snow leopards actively targeting domestic livestock over wild game? Gut contents alone cannot answer that. A high rate of consumption might just reflect sheer environmental availability rather than an evolutionary preference. To separate preference from abundance, the research team generated a relative abundance index by running over 4,500 camera trap nights to map the exact prey baseline of the mountains. The camera traps recorded a staggering density of domestic livestock occupying the same high-altitude zones as the predators. By contrast, there is a relative scarcity of wild ungulates, like blue sheep and Himalayan tahr, traversing these slopes. Researchers then ran a statistical selection analysis, comparing the snow leopard's diet directly against this baseline abundance index. The resulting standardized preference indices resolve the paradox. Relative to their massive abundance, snow leopards actually avoid hunting yaks. When given a choice, snow leopards heavily prefer wild prey, actively selecting blue sheep and tahr whenever they are available in the environment. The high caloric consumption of livestock is an opportunistic artifact of a severely livestock-dominated landscape, not a biological preference. This data solidifies the snow leopard's verified ecological profile. It is a highly plastic generalist capable of vast dietary shifts depending on what its territory offers. This plasticity manifests across seasonal and regional boundaries, forcing the cat to constantly adjust its reliance between wild prey and domestic herds. However, even relative biomass calculations have a lingering methodological blind spot. The equations rely on the average body weight of adult animals. Adult yaks are large and difficult to kill. Snow leopards often prey on lighter, easier-to-catch juveniles, meaning the current metrics likely overestimate the total caloric yield of livestock. Zooming out, this points to a broader ecological crisis across Central Asia, where the biomass of wild herbivores has plummeted to less than 5% of total livestock biomass. Mathematically, the most critical intervention to restore ecological balance is increasing the population density of wild ungulates. Restoring wild prey draws these predators away from farms, but their opportunistic nature means livestock predation will persist.